This is a chapter that will be fairly large because it's going to cover not only the individual nerve cell, but the anatomy of the nervous system. We discussed this a little bit in an earlier chapter when we discussed tissues. Nervous tissue is composed of two different types of cells. Nerve cells are known as neurons, and they are packed and sustained by support cells known as glial cells, or sometimes simply referred to as glia. Neurons are the cells which will use electric current to communicate with other cells. They communicate with other nerve cells, they communicate with glands, and they also communicate with different types of muscle cells. These cells are known as irritable or responsive because they react to a stimulus. Neurons are among some of the few cells in your body that do not reproduce themselves. There's only a small number of cells in the brain that will do that. That's actually in the hippocampus, and those weren't discovered to undergo neurogenesis until about a decade ago. Glial cells are also referred to as glia, or simply neuroglia, and these again are the support cells. Some act as phagocytes, cleaning up damage and debris. Some act as insulators, and then others will actually protect the nervous system from anything that might be invading the body through the bloodstream. Unlike neurons, the glial cells will reproduce themselves, and they are responsible for much of the growth that occurs in the size of the brain and the spinal cord throughout the life. Also, when an adult has brain cancer, it is almost invariably going to be one that's derived from glial cells. You'll hear people talk about a astrocytoma or a glioma, something like that. You won't hear about tumors derived from neurons, such as neuroblastomas, except in babies and children. I like to make a flowchart showing the divisions of the nervous system. And the first level would be separating the central nervous system from the peripheral nervous system. The central nervous system is composed entirely of the brain and the spinal cord, and the peripheral nervous system is basically everything else. The peripheral nervous system can be further divided into the sensory division, responsible for bringing in information from the environment, be it internal or external, and then the motor division, which has the job of sending information out to glands and muscle cells. Terms to be further associated with the peripheral nervous system would be the sensory system is referred to as the afferent system, and the motor system is referred to as the efferent system. What these words refer to is basically incoming and outgoing. So afferent is sensory information is coming into the central nervous system, and then the motor system is the efferent, or the outgoing instructions from the central nervous system. The motor nervous system can be divided into the autonomic system or the somatic system. I always tell students when you hear the term autonomic, you should think automatic. The autonomic system, sometimes referred to as the visceral nervous system, is totally involuntary. You've got no control over these nervous impulses. These nervous cells will control the smooth muscles, which you'll find around the hollow organs such as the respiratory tract, digestive tract, reproductive tract, urinary tract, and also the cardiac muscle cells obviously found in the heart. The somatic nervous system is voluntary, and it controls the skeletal muscle. And then finally, the autonomic nervous system is divided into the sympathetic system, which is your fight, fright, flight, and freeze, and your parasympathetic system, which is your rest, nest, and digest. In other words, the sympathetic system is going to be the system that controls functions that would help you escape a danger. It's going to increase your heart rate, the force of cardiac contraction. It's going to dilate your lungs, the passageways in your lungs, so you can get more oxygen to escape. It's going to increase your blood sugar, so you've got more energy. It's going to decrease your immune system or your inflammatory response. And then the parasympathetic system is going to be the one that's going to relax you. It's going to be in control. For example, when you're simply resting and vegging at home in front of a TV. It's going to allow you to be hungry. It's going to increase your digestive processes, and it will allow you to become romantic. It's going to allow things like male erection and female arousal. So I'm going to fight with the stylus to try to show you what I'm talking about when I say a flowchart. So the nervous system is going to be divided into the central nervous system, which includes the brain and the spinal cord, and then you've got the peripheral nervous system, and that's going to be divided into the sensory side and the motor side. And then the motor is divided into the somatic nervous system, that's the voluntary system that controls the skeletal muscle, and then the autonomic system. And then the autonomic system is divided into the sympathetic system and the parasympathetic system. So forgive the writing, but you get the idea, and my brain works better when I see things organized in a flowchart type material. So to complete some definitions for you, first off, I've already told you that the sensory side of the peripheral nervous system is going to be bringing in information both from your internal environment, things like blood pressure, acid base, electrolyte status, and then also information from the external environment, things like something you might hear, something you might smell, your ambient temperature. So these are going to be detected by cells known as receptors. And you'll have generalized receptors, which we'll discuss later, all over your body that might pick up something like temperature. And then you'll have specialized receptors, things like taste, hearing, sight, etc. Again, these will be further discussed in later chapters. But most receptors are activated by changes. In other words, you won't notice something until there's a change in status. And at the end of the neurons from the motor side of the peripheral nervous system, you'll have what's known as an effector. Remember, the efferent system is your motor system. And the effector is going to either be a gland or a smooth muscle cell or cardiac muscle cell if we're talking about the autonomic system, or it's going to be skeletal muscle if we're talking about the somatic system. So let's take a closer look at nerve cells themselves. Again, these are referred to as neurons. First off, the soma is a cell body. So the soma is going to be this region right there. Dendrites are the branches that will bring information into a neuron. So these receive input. And then axons are going to be the extension that's going to carry information away from the cell body. Just a little factoid here, a cell can have thousands and thousands of dendrites, but it will always only have one axon. Something that's important to understand is that, except for a very few circumstances that we'll discuss, nerve cell bodies are going to be located in the central nervous system, and the peripheral nervous system is going to be composed of mostly axons. So when we get further into this, I'll talk about that a little bit more. But when you refer to, for example, somebody says, oh, I cut a nerve in my arm, actually what they did was they cut an axon. And sometimes these will actually repair themselves pretty decently. If you actually have a spinal injury or a brain injury, then you may have destroyed the soma, and those won't come back. So the fact that the nerve cell body, or the soma, is in the central nervous system, and the axons carry information to things, think about your muscles of your big toe, that means that if you were to try to draw these things from scale, it'd be, or to scale, it'd be very difficult. Because think about, you've got this tiny nerve cell body in your spinal cord or in your brain. So the axon for this neuron can be potentially a couple of feet long. Think about it, even 3 feet long, depending on your height, because it's going to be going from the spinal cord down to your big toe. We discussed the organelles of a stereotypical cell in the earlier chapters. There are a few things that are termed differently when you're talking about neurons. First off, the neurolemma is the same thing as the cell membrane. Another word for cell membrane is plasmalemma, so that's simply the cell membrane. And then the cytoplasm is going to be referred to as the perikaryon, peri meaning around, karyon meaning body. So I don't know really why they only refer to that as neurons, because the karyon is referring to, again, your body of your nucleus, which is going to contain your karyotype or your chromosomes, which would be true for any cell. But anyway, we only use this term for neurons. Something else that's unique to neurons is the presence of something referred to as Nissl bodies. Nissl bodies are clumps of rough endoplasmic reticulum and ribosomes, and they are found in the soma of the neuron. So these are very distinctive, and they cause the nerve cell bodies to have a gray appearance. For this reason, nerve tissue that has nerve cell bodies in there, for example, the cortex of the brain or the inside part of the spinal cord, is going to have a gray appearance and is referred to as gray matter. So I mentioned to you earlier dendrites. These are going to be the very fine little processes that cover the surface of the neuron, and these bring information into the cell body. And there can be tens of thousands on a single neuron. Axons, again, these will be carrying the information, which is going to be carried by the action potential. We discussed that when we talked about muscles. This is going to be carrying information away from the cell body to transmit it to another neuron or a muscle cell or a gland. You'll see some different terminology in your reading. They refer to the cell membrane around the axon as an axolemma, and they refer to the cytoplasm as the axoplasm. I apologize, by the way, for the sound effects being contributed by my dog. Axons have a couple of regions I wanted to mention because they'll come into play a little bit later. First off, the axon hillock is the little butt of the axon as it leaves the soma. So again, information will be transmitted in this direction. And then at the end of the axon down here, you'll find what's referred to as a telodendria, and they end in a synaptic terminal. So it would be this. So the axon will be this direction. Again, if I were to draw an axon, I would draw branches. These branches would be the telodendria, and then the synaptic terminals would be on the end of that. Sometimes you'll see it referred to in some textbooks as synaptic buttons, terminal buttons. There's a myriad of names, but they all pretty much mean the same thing. And the term I tend to use is synaptic terminal. And the reason for that is where these neurons meet another cell, that space is going to be referred to as a synapse. So if this is cell number 2, then this region is a synapse. Another special thing about axons is sometimes they have a white greasy wrapping around them referred to as the myelin sheath. So the myelin sheath acts as an insulator that prevents the action potential from being dissipated throughout the tissues, and is produced by, in the peripheral nervous system anyway, by a specialized cell type known as a Schwann cell. Here's a nucleus, and as you can see, it just wraps around and around and around the axon. This myelin will give these axons in the peripheral nervous system and in the central nervous system, although there's not as many there, kind of a white appearance. So as tissue that contains the nerve cell bodies, the somas, which contain Nissl bodies, are referred to as gray matter. When there's a lot of myelinated axons in a tissue, it's referred to as white matter. And we'll discuss that location a little bit later when we start talking about particular anatomy. So myelination is important because it's going to make the action potentials be able to travel faster. The reason that happens is because of the entire axial lemma having to be depolarized as the action potential flows toward the synaptic terminals. It will only have to hop from gap to gap. So these gaps are referred to as nodes of Ranvier. So the passage of this action potential is referred to as propagation. And propagation will be very much faster on a myelinated axon than a non-myelinated axon. So when your body needs to transmit information critically fast, it will use myelinated axons. If the information is not so urgent, let's say it goes to, let's say, a thyroid gland or something where it's going to act over days to weeks, it's going to use a non-myelinated axon. I mentioned this term earlier to you, the synapse. So a definition is an area where two cells will communicate. And in particular, what we normally talk about will be a neuron that's going to be transmitting an action potential, or not transmitting, but having an effect on a postsynaptic cell. So neuron number 1 would be your presynaptic cell, and it's going to be communicating with a postsynaptic cell. And the vast majority of synapses in your body will have what's referred to as a synaptic cleft, which is this gap between these two neurons. There are a few specialty types of synapses that do not have a synaptic cleft, but we'll mention those later. Let's look at the components of a synapse. Again, I've already mentioned to you the synaptic terminal, and that's going to be the whole bulb that you see here at the end of the teledendria or axon. And the synaptic terminal is going to contain these little envelopes known as synaptic vesicles. They contain a neurotransmitter. A neurotransmitter is simply a chemical that's going to have an effect on the postsynaptic cell. You'll see on this diagram that you have receptors for that neurotransmitter. And so that's how cell number 1 will have a reaction on cell number 2, because the neurotransmitters will be released and bind with the receptors and cause a reaction. So just to give you a little perspective, again, these are really not drawn to scale at all, but here you'd have neuron number 1, and you see all the dendrites that are getting information into the cell. If it's going to be enough to cause an action potential, that action potential will be transmitted down this axon to neuron number 2, and you see these various synapses. And again, here's a close-up of a synapse, and you can see the synaptic vesicles containing the neurotransmitter being released, and it's binding to the receptors on the postsynaptic cell. There are several types of neurons, but I just wanted to mention 2 types in particular. First off, you should know that the most common type of neuron in your body is going to be referred to as a multipolar neuron. And what you've got in this arrangement is you have the collection of dendrites, and then you have the one axon at the end. So the other one I wanted to point out is called a unipolar neuron. And what happens is you've got one process that comes out, and it then divides up into axons on each side. So I'm pointing this one out because the sensory neurons in your body are going to have this arrangement. And we'll be talking about sensory ganglia, and you'll see that you've got input coming in, and it's going in on an axon, going through the cell body, and then continuing on to the spinal cord. So I had a student ask me one day, well, why do you refer to axons when you talk about sensory neurons when you're actually picking up information from receptors? And this is why, because we're talking about unipolar neurons. We can also classify neurons based on their function, and I've already mentioned to you sensory neurons, also referred to as afferent. I've mentioned to you motor neurons, also referred to as efferent, but there's a huge pool of neurons between these two that are very important, and I want to mention those. Those are referred to as interneurons and also called association neurons. So these actually, again, are going to be intermediate between these two, and they have the job of analyzing information that's being brought in. They help you decide something, they help you remember something. So this is actually the largest class of neurons that you have in your body. There are three types of sensory receptors that I want to give you the definitions of. First off, interoceptors, and just like it sounds, these monitor internal systems. So these will pick up information, let's say you're hungry, or you're maybe having chest pains, or you need to urinate. There are some senses that will be classified as being from interoceptors. Taste, pressure, and pain are examples. Exteroceptors, again, just like it sounds, these pick up information from the external environment, things like sight, smell, hearing, and temperature, and pressure that's simply not a deep pressure. And then finally, the third category of receptors will be the proprioceptors, and these are going to be bringing information about your body position and movement to your central nervous system, and they give you a spatial awareness. You know without looking the position of your feet relative to each other, or of your hands relative to each other. These do things like help you climb stairs without watching your feet. They help you sit upright. These will be giving information to and from your postural muscles. And these are receptors that are located inside your joint capsules and also in some of the skeletal muscles. The smallest group of neurons that you've got, I've already mentioned that you have a huge group of interneurons. Sensory neurons, that's your second largest group, and then your smallest group will actually be the motor neurons. And again, these have an effector with an E, and they're going to be sending information to muscle tissue or glands. The interneurons I mentioned, again, these are actually going to be what separates us from lower life forms. They let us do things like analyze and memorize and plan and learn and make executive decisions. They're taking the information from the sensory neurons and deciding whether or not to send the information to your motor neurons. These next few slides will talk about the glial cells, and these are over half the volume of the nervous system in the average adult. Let's first look at the central nervous system, and it's an important distinction. These are only in the brain and the spinal cord. The first type is an ependymal cell, and these are in the cavities of the brain, referred to as the ventricles. And there are clusters of specialized capillaries, little tufts of capillaries called the choroid plexus. There's an example right there. And these ependymal cells that are found there will filter out plasma to create cerebrospinal fluid. That's a fluid that's going to bathe and cushion the brain and the spinal cord and provide things like oxygen and nutrition. The second type of glial cell in the central nervous system is called the astrocyte. And these cells have the job of providing a barrier between the nervous system and the bloodstream. This blood-brain barrier is vital because it's going to prevent average blood infection from becoming an automatically fatal central nervous infection, but also it's going to have to be considered when you're choosing anesthesia, and also if you're trying to find an antibiotic or some other type of medication that you need to penetrate into nervous tissue. So here's a closer look at what I'm talking about with the blood-brain barrier. On this side, you've got the brain and the cerebrospinal fluid. On this side, you've got the blood system. So your astrocytes are going to help provide one more barrier that things would have to pass through to get into the central nervous system. In the central nervous system, the cell known as the oligodendrocyte will form the myelin sheath around the axons. Please note these are only in the central nervous system. They are called an oligodendrocyte because you have one cell that's going to provide the myelination for several different neurons in the CNS. Another type of glial cell in the central nervous system is referred to as microglia. These are macrophages or phagocytes, remember a cell that eats. These are the janitorial staff cells of the central nervous system, and their job is to clean up any sort of waste, damage, or any pathogenic organisms. Now, switching away from the central nervous system to the peripheral nervous system, there's only two types of glial cells. The first one is a satellite cell, and they're called that because they surround a ganglion. I mentioned to you earlier that all nerve cell bodies are located in the central nervous system except for an exception, and the exception is a ganglia. A ganglion is a cluster of neuron cell bodies. In particular, we'll look at that with the autonomic nervous system. For example, if this is the spinal cord, then the ganglia are going to be located around the spinal cord. So the satellite cells will surround these ganglia and provide protection. In the peripheral nervous system, the myelin sheath is going to be created by a cell known as a Schwann cell. With the oligodendrocytes in the central nervous system, we said that one cell is going to be myelinating several different axons. Well, here in the peripheral nervous system, one axon will actually have a series of Schwann cells that are creating the sections, the segments of the myelin sheath that are separated by the nodes around VA. One of the important jobs of the myelin sheath, especially in the peripheral system, is the ability to allow the neuron, the axon itself, rather, to heal after an injury. So if you picture that this would be in the spinal cord, and then this is going to be your peripheral nervous system, so let's say this is an axon that's going down your arm. If you accidentally cut that, what's going to happen is, first off, there's a process known as Wallerian degeneration where the axon actually disintegrates. So to heal, the Schwann cells will actually regrow down the side of that injury, and it forms what's known as a regeneration tube that provides a pathway for the new axon to grow. This doesn't always happen. Sometimes you end up with just a permanently dead axon, but at least in the peripheral system, there is a possibility of healing. If you have an axon that's damaged inside the brain or the spinal cord in the CNS, it may or may not heal because you don't have the Schwann cells. You've got the oligodendrocytes who are actually tending to a lot of different neurons at one time. So the healing process, if it does occur, is going to take a whole lot more time. So again, if you have damage to a nerve cell body, then the nerve cell bodies cannot heal. If you have nerve cell bodies die to an injury such as a stroke, what will happen is if those cells are dead, they stay dead. Some people can heal very well from a stroke because the brain can actually rewire circuits around that injury to take the place of the cells that are no longer there and functional. It becomes basically a process of rewiring the brain. Spinal injuries don't do nearly as well, and that's why, again, you can come back from a stroke, whereas if you have a severe spinal injury, typically there is not a healing process. So now let's take a look at how the information is going to be transmitted down neurons. It's transmitted by electric current, and if you recall back in earlier chapters about the cell, I mentioned the fact that the inside of the cell is going to have an overall negative alignment of charges next to the cell membrane, whereas the outside of the cell will have an overall positive alignment of charges. These are produced by the electrolytes, in particular sodium and potassium. So this is the outside and this is the inside. And this creates what they refer to as a transmembrane potential. That means that there's a difference in the electric voltage on the inside versus the outside. So for this to happen, there are several things that must be in place. First off, you have to have a concentration gradient. A concentration gradient is defined where you have a high concentration of something in one area and then a low concentration on the other area, what's going to happen is these ions, if they have a chance, and let's say you've got a membrane that has a large opening and there's no problem with these ions passing through, these are going to go through that opening and will equilibrate until there's roughly an equal concentration on each side. Now what happens if their opening is too small or it doesn't exist, then what's going to happen is the ions simply will be separated and they cannot diffuse. So let's say that there is a gate that covers this and we've got a separation of charges. Now let's say there's some sort of signal that will make this gate open up, and so in other words this becomes open, then these ions are going to go ahead and diffuse until you have an equal concentration across. So what they do with this is they have channels that are gated and channels that are always open. We'll talk about the two different types of channels. And these are the openings in which the ions sodium and potassium can move through. There's a term, if you recall from earlier chapters, selective permeability. It means some things are able to pass through freely, some things are regulated. So when there is a change in the permeability, when I use the term permeability, I'm talking about the ability of an ion to move across that membrane. And please remember, when I say ion and electrolyte, those things are interchangeable. It's the same thing. And the ones in particular we're talking about is potassium and sodium. So when the transmembrane permeability changes, the transmembrane potential will change. And this happens, you're changing the permeability by opening or closing a channel. There are two types of channels. There are passive channels and there are active channels. Passive channels are always open. Sometimes you'll see them referred to as leak channels. And you simply have ions that move across there by random diffusion. The active channels, also known as the gated channels, they have to be triggered to open. And what triggers them is going to be that electric current, the action potential that's being transmitted down that membrane. So this is how that electric current is being transmitted down the membrane as it goes. It's opening and closing particular gates in a particular sequence. And it's allowing, when a gate is open, again, picture you've got a cell and you've got the alignment of the negative charges along that inner membrane. You've got the positive aligned on the outside. The outside of the cell, your interstitial fluid, has a lot of sodium and a little bit of potassium. The inside of the cell has a lot of potassium and a little bit of sodium. So again, picture these as particulates. So if you've got gates that are particular for each ion, in other words, you have to have a potassium channel that's going to be allowing only potassium ions to go through, or you've got to have a sodium channel that's going to only allow sodium to go through, something's going to trigger these to open. So let's draw our little gates in here. And let's say the red will be the sodium channels. When they open up, now they're open, what's going to happen is sodium is, because you've got a lot of it out here, it's going to move down the concentration gradient by diffusion into the cell. Then you're going to have openings of potassium channels, so let's draw those in here. Now they open, and so potassium, because you've got more inside the cell than outside the cell, you're going to have potassium that's going to move to the outside, down its concentration gradient. So what I have in this diagram, these are actually pumps that run using energy, so therefore this is active transport. These exchange pumps will actually reverse this process. After the signal has been transmitted and the action potential is over, you've got to repolarize this And so you've got to put the sodium back out of the cell, and you've got to put the potassium back inside the cell. And that happens through active transport through the sodium. In fact, let me go ahead and write that right here because it's not printed on the side. The sodium potassium exchange pumps. So we'll discuss those a little bit more in future slides. So again, the leakage channels, these are the ones that are not gated, and they just will randomly allow the electrolytes, or the ions as they're calling them here, same thing, to diffuse across. They're going to be diffusing down the concentration gradient from an area of high concentration to an area of low concentration. Again, there is no signal being transmitted here. It's simply helping maintain the concentration gradient. The ones that really do the job are the gated channels, and they have to get a message. You have to have a trigger to open these. And the word ligand simply means a chemical molecule. And in particular, what we're talking about here is a neurotransmitter. And so if you see this is a neurotransmitter, and here it's binding to a receptor on this channel. And once it binds, it's going to cause this gate to open over here. And now you're going to allow the ions again to move down the concentration gradient. So you'll have sodium again moving inside the cell if this is a sodium channel, or if this were to be a potassium channel, you'd have potassium moving to the outside of the cell. Some channels use simply a mechanical gate. You'll see these when you look at some of the receptors in your fingertips, for example. The sense of touch, a lot of these are mechanically gated receptors. Also, hearing and equilibrium, these use mechanoreceptors. And what it means with a mechanoreceptor is that the gate is triggered by mechanical deformation. And you simply, once there is a deformation in the receptor, it's going to cause the gate to open, and again, then you've got the ion movement. Another example would be a voltage-gated channel. And these are the ones that we're talking about when we're talking about transmission of an action potential down a neuron cell membrane. So let's look at a chart. Now I've told you the definition of a transmembrane potential, and that's going to be assuming then that we have a level of voltage, and it's typically a negative level, usually it's negative 70, sometimes you'll see it negative 90, that you have a resting membrane potential. The resting membrane potential in this cell is going to be at negative 70. So if this cell is sitting here at negative 70, it's at resting membrane potential. It's not doing anything. But now what happens if we have a stimulus, and the stimulus is going to be occurring right here, for example, and that's going to cause a change. When you have a stimulus applied, it's going to cause those channels to open. You're going to have the ions move across the cell membrane, and now we've produced a change in that transmembrane potential. So if it is a small change that does not reach threshold, which is right here, it's simply referred to as a graded potential. A graded potential is just a small temporary change. It's localized. It just kind of fades away the further you get away from the stimulus. However, if it does reach all the way to threshold, now you're going to produce an action potential. So an action potential is going to be produced by a very large graded potential or a collection of graded potentials, and it actually is propagated or sent along all the way down the axon. It does not change in strength. I always tell my class it's like being pregnant or not pregnant. You either have an action potential or you don't. You have to be able to reach this very important threshold. Now here's the pump I mentioned earlier. So again, we've had an action potential. We've had the ions switch down the concentration gradients using, again, passive transport. So now we've got to restore that to make the cell ready to be fired again, so we can use it again. And these sodium-potassium exchange pumps are going to be trading sodium, so we're moving sodium back outside the cell and moving potassium inside the cell. And this is what maintains that resting membrane potential. But the trick is, this is active transport. It requires energy. So without oxygen, without energy, you're not going to be able to maintain the nervous system because you cannot restore that resting membrane potential. So be sure to understand, again, this is one of the big reasons that we have to have oxygen, and that we're basically brain dead within just a few minutes of not having oxygen, is because without oxygen we cannot manufacture energy. And again, when you see the term ATP, you should be thinking about energy automatically. And ATP is what drives the sodium-potassium exchange pump. So without oxygen, you have no energy. Without energy, you don't have functioning neurons. So let's take a closer look at what happens with action potentials. And to do that, we're going to have to look at the term depolarization. Depolarization is the term used when you're becoming more positive. Again, look at this diagram. Here is your resting membrane potential. You're at -70. Here is 0 right here. Here's a threshold. And let's get rid of that term graded potential out of the scenario right now because I don't want to confuse the issue. We have a stimulus. Now you've got those channels opening up in a particular sequence. And what you're going to see in just a minute, the first thing that's going to happen is you're going to have the sodium channels open, and that means sodium is going to be allowed into the cell. Now, this -70 is, again, that's the resting membrane potential, and we're going to be adding positive ions, these sodium, positively charged sodium ions, and that's going to move this membrane potential in a positive direction. You shoot right past 0, now you're at +40. So here is a peak of your action potential. Again, you've become less negative, and that's important to understand. So that is depolarization. I'm going to walk you through this step by step, but what happens after is then you go through repolarization, where you become more negative again, and then you're going to actually go through a period of hyperpolarization where you're down below resting membrane potential, and then finally return to resting membrane potential. Now, if you're talking about a graded potential, you'll still get a depolarization, but you're not going to go past threshold, and so what's going to happen is the transmembrane potential will simply return to the resting membrane potential. In other words, you don't hit threshold, so no action potential is going to be produced, and it will simply fade away. So if you do reach threshold, now you've produced an action potential. So again, what's going to happen? You're going to have the application of a stimulus right here. You're going to start opening sodium channels. That's going to allow sodium channels to go into the cell. And now you've changed the voltage on the inside of that cell membrane. And this is your depolarization. That's your sodium channels over here. So moving into repolarization, what's going to happen is you're going to have an inactivation and then a closing of the sodium channels. And now you're going to have an activation and turning on of the potassium channels. So it's going to be the potassium channels that are most important over here. Again, down here, it was sodium. But now we're moving the potassium into play on the repolarization side. So after you've repolarized, the potassium channels will start to close as soon as it's reached normal resting membrane potential, and you move into a phase known as hyperpolarization. So again, if you think of your chart, what you've got resting membrane potential, you've got depolarization, then you got repolarization. Now you're at resting membrane potential, but you continue downward, you've gone into hyperpolarization, then you return back to resting membrane potential. So be sure to understand that in a hyperpolarized state, you're even more negative than you started. So instead of a resting membrane potential of -70 millivolts, now you've reached -90. As I mentioned earlier when I said you either have an action potential or you don't have an action potential. I said this is like being pregnant, either or you aren't. This is known as the all or nothing principle. So again, if you've got a graded potential will simply fade away. However, if you have an action potential, it's going to reach threshold and it will always be the same strength. You don't have a large action potential or a small one. There's something known as a refractory period. I mentioned this just very briefly when we talked about trepa formation and wave summation, when we talked about muscle contraction. And the reason that cardiac muscle is different from skeletal muscle as far as the way it acts, with skeletal muscle, again, you can start a stimulus and you can keep on going in more and more and more and more. But with cardiac muscle, you always have that nice regular rhythm. And that is because of something known as the refractory period. So during this absolute refractory period, you cannot stimulate another action potential no matter how hard you try. Now during hyperpolarization, you've got a relative refractory period that if you try hard enough, then you might be able to stimulate another action potential. So when I talk about a refractory period, that means something is either more difficult or impossible to do. I talked about the word propagation on just a slide or two when we talked about myelination of axons. So let's look at the significance of myelination on these axons. And again, propagation is simply sending or transmission of that signal down an axon. And what's happening as it goes down is you've got the progressive opening of these channels as it goes down the axon. If an axon has no myelin, in other words, if it's unmyelinated, then you're going to simply have the channels open and close. If this is the axon, you've got to transmit, you've got to open every little channel along the way. And so, again, I've mentioned the inside being more negative in relation to the outside. You've got to have the electrolytes that are going to switch all the way down the axon. So it's got to be continuously down that axon. Saltatory propagation, and that's what this example is, occurs when you have myelinated axons. Remember that in the peripheral nervous system you have Schwann cells, and you have many of them that will wrap around these axons. And these gaps are referred to as nodes of Ranvier. So in an unmyelinated axon, you've got to open all these channels to propagate that action potential. But in saltatory propagation, the impulse can actually hop from node to node to node. So you're only having to do the depolarization or repolarization at the nodes. So this is much faster. So make sure you understand continuous propagation is on non-myelinated axons, and it's slower, and saltatory is on myelinated axons, and it's much faster. And the reason that saltatory is faster is simply because, again, you only are having to depolarize at the nodes of Ranvier. There's another aspect that's going to affect how quickly information will be transmitted down a neuron axon, and that's going to be the diameter. Think about the diameter of a water hose. If you have a very narrow, very small water hose, then the water is going to have a lot of friction up against the sides of the hose. So if you have a very large water hose, then there's a whole lot of water that's not going to be coming in contact with the walls of the water hose. So it's going to come out much faster. So, a fire hose has a very, very large diameter, and the water is able to come out very quickly. You can take the paint off a wall, it comes out with such force. And an axon is the same way. So if you've got a very small diameter axon, it's going to move very slowly in comparison to the very large diameter axons. And then when you add on to that myelination, then indeed you've got the fastest propagation. So a very small unmyelinated axon is going to give you the slowest propagation, and a very large diameter myelinated axon will give you the fastest. If you do the math, the propagation speed of a large myelinated axon will be along the range of 228 miles an hour, whereas a non-myelinated small axon will be more along the range of 2 miles an hour. I'm going to describe some different types of neurons and try to explain why the speed is different, why we don't need necessarily a very fast transmission down these really small non-myelinated neurons. I really don't care that you learn A, B, or C, but we'll just walk our way through them. I do want you to understand, again, if I were to say which one of these neurons is going to have the fastest transmission, I would expect you to be able to tell me it's going to be the largest diameter myelinated axon. These fastest neurons, again, we're going to be carrying information very quickly to keep you upright. It's going to help you maintain posture and balance, and also it's going to help you respond reflexively very quickly. Your medium range neurons, these are going to be myelinated, but their diameter is not quite as large. These will be carrying information coming from your special senses, also from proprioception and also from general senses such as touch and pain and temperature. And then finally, again, these small fibers that are non-myelinated, these are going to be very slow, and these might be involuntary muscle contractions in your digestive tract, or again, glandular controls. Now, I defined a synapse for you earlier as basically a place where two cells communicate, and normally we're talking about a neuron communicating with another neuron, or a muscle cell, or a gland. For our purposes for the next few slides, we're going to be talking about neuron-to-neuron synapses. I apologize for my messed up slides. I've noticed several in this presentation that this app has squashed together, but I can't fix it now. But anyway, the first of the two types of synapses is not as common. These are electrical synapses, and they are unique because they actually have something known as a gap junction, which allows the ions, such as sodium and potassium, to go straight from cell to cell. The purpose of these is to allow the continuous propagation of an action potential so there's not a synaptic delay. So you'll find these in areas where communication is absolutely critical, not to be slowed down by a synaptic delay, areas of the brain, eye, and ear, and ciliary ganglia. By far the most common type of synapse you'll have in your body is called a chemical synapse. With these, unlike with the electrical synapses, which actually you have cell-to-cell contact, these cells don't actually touch. So what you're showing here is this is going to be your neuron number 1, and that's your presynaptic cell, and here is your postsynaptic cell. So the information will be coming down this pathway. So I would normally say that the action potential is going to go from the presynaptic cell to the postsynaptic cell, but there are some circumstances where the information that's going from the presynaptic cell is actually going to inhibit the postsynaptic cell. So there are some times where the information that's being passed is going to tell the postsynaptic cell not to be activated. So that would be a type of inhibition. We'll talk about that in just a second. So there's going to be some variation in the information passed, and it's going to depend on the type of the neurotransmitter, which is going to be located in these little vesicles. And it's going to be depending on the sensitivity of the receptors that you'll find in the postsynaptic cell. So remember what happens. You've got a neuron that's going to be up here somewhere. You've got the axon coming down. You've got the transmission of the action potential that's going to actually trigger calcium ions, which we really haven't talked about those, so don't get too hung up on that. But they will enter the synaptic terminal, and that will cause the synaptic vesicles to dock and release the neurotransmitter into the synapse. So remember, there is a synaptic cleft in these chemical synapses. One of the things to understand about a synapse is that the effect of the neurotransmitter is going to depend on the receptor and not in particular the neurotransmitter itself. And for example, the neurotransmitter acetylcholine can actually stimulate an action potential at the neuromuscular junction that we talked about in the skeletal muscle, but it's actually inhibitory at the neuromuscular junctions of cardiac muscle. So that is all going to be based on the type of receptor that you have. When we talk about chemical synapses, there are two particular types I want to mention. The first one is going to be a cholinergic synapse, which we'll talk about on the next couple of slides, and then we'll talk about something called an adrenergic synapse. Very simply, the cholinergic synapses all use the neurotransmitter acetylcholine. These are going to be all the neuromuscular junctions in the skeletal muscle system. We do have some cholinergic synapses in the brain and the spinal cord, and there are some in the peripheral nervous system, but we'll be talking about them more when we start talking about pre- and post-ganglionic neurons in the autonomic nervous system, and that's a different chapter. When an action potential is passed from a presynaptic neuron to a postsynaptic neuron, there's going to be a very tiny delay. We're talking tenths of a millisecond between the arrival of the action potential to the transmission of the action potential or the effect on the postsynaptic membrane. This is because it takes some time for the docking of the vesicles, the synaptic vesicles. It takes time for the release of the neurotransmitter. It takes time for the binding of that neurotransmitter to the receptors. So this is a much faster process than what we can possibly even imagine, but it is still a measurable period of time. So you do not have a synaptic delay in the electrical synapses, but you do in the chemical synapses. So this leads us to a concept that the fewer synapses in the pathway that you have of an action, you're going to have a faster response. So the more synapses you have, you're going to have a slower response. So if you start thinking about reflexes, reflexes are these pre-programmed automatic responses that we have. Quite a bit of them are pretty involuntary. We can mentally overcome some of them. Think about putting contact in your eye or putting drops in your eye. You have an automatic corneal reflex that you have to master and overcome. But you respond quickly to avoid being physically harmed as a biologically programmed thing. So these reflexes will have very few synapses, so they happen very quickly. However, fewer synapses means that you've got a more predictable outcome. And that's what you'll see in what they consider to be less evolved forms of life. And I use that with with a little bit of worry. But in lower levels of animals, you'll have a faster reflex, but you have a much more predictable response. Again, think about an animal such as a chicken or a lizard or something like that. There's just not that many alternate reactions you're going to get. So understand again that fewer synapses means faster response but more predictable, whereas more synapses will be a delayed response, but you've got more possibility of a variable outcome. So what's going to happen after an action potential is the leftover acetylcholine will be metabolized out. There is an enzyme called acetylcholinesterase that will help break that down, and parts of it will be taken back up into the axon by axonal transport, and it'll be recycled. So again, all this does take time, and if the stimuli keeps coming faster and faster and faster, and so in that synapse, the demand for neurotransmitter cannot be met. In other words, you can't recycle that neurotransmitter fast enough. You're going to run out of acetylcholine. You'll actually end up with synaptic fatigue. So think about constant stimuli and running out of acetylcholine and not being able to respond to a stimulus fast enough. And if you've ever been acquainted with drowsy driving, it's actually, they say, possibly equally or even more dangerous than drunk driving. That, I think, would be a good example of synaptic fatigue. There are over 100 neurotransmitters, or discovering new ones every year. There are two general classes. Some will cause the postsynaptic cell, the neuron number 2, to depolarize. That means these are considered excitatory neurotransmitters. If you hyperpolarize that postsynaptic cell, that's considered an inhibitory neurotransmitter. Remember we talked about, again, picture your chart. You've got your resting membrane potential, you got a stimulus that arrives, now you got depolarization, now you got repolarization, now you've got hyperpolarization. And think about it, if you're at a hyperpolarized state, it's going to take much stronger stimulus to actually stimulate a second action potential. It's going to be less likely to refire. So let's throw in our, well, that's a pretty bad straight line, but you kind of get the idea if that were to be our threshold. So understand that excitatory neurotransmitters will cause neuron number 2, the postsynaptic neuron, to depolarize. You're going to cause an action potential. But an inhibitory neurotransmitter is going to hyperpolarize the membrane of the postsynaptic cell, and that's going to inhibit further action. Now, a lot of times you think of inhibition as being a bad thing, but there are parts in our brain, especially not the basal ganglia, this inhibition is critical because it will smooth out a movement. There are circuits in your brain that would direct your hands, for example, to reach something, but it would be a very hard, jerky movement, but the inhibitory neurons will smooth those out. Or, for example, if you need to urinate, you have to inhibit the neurons that are signaling the sphincters to close. So you have to turn those off so you can actually pee. So I told you there were two classes of neurotransmitters that I was going to talk about. And I mentioned cholinergic synapses. Those are the chemical synapses that use acetylcholine. This is the second type, adrenergic synapses. These use neurotransmitters known as catecholamines, and the big ones that we talk about are norepinephrine and epinephrine. The older term for epinephrine was adrenaline, and that's where the verbiage comes from. Another one is the neurotransmitter dopamine, and this will be a kind of a mood elevator, and they've used this to treat Parkinson's disease. And they've also discovered that the reason cocaine is addictive is that it mimics the sensation that you get with dopamine stimulation, and it's why you feel this hyper-attentive, energetic ability to jump off tall buildings. Another neurotransmitter I wanted to mention was serotonin, and this has to do with relaxation, and you may have heard them advertised on television. Antidepressant drugs, you'll say this is an SSRI. What that stands for is selective serotonin reuptake inhibitor. So think about it. You have a neuron that's going to be releasing serotonin, that's going to be affecting the receptors on the postsynaptic neuron. And serotonin is in the synapse and is causing all these nice mellow conditions here. And normally you would have enzymes that's going to break that down. However, you have added now a reuptake inhibitor, so you're going to block the reuptake of the serotonin basically. So now you're going to keep the serotonin in the synapse. So you're going to enhance this mellow mood. So that's why this is used for so many antidepressants. The problem is these are very addictive drugs. I've known, in fact, I had a young woman that worked for me that was taking a really, really high dose of Paxil. She was self-medicating, and she ended up losing her career over it, and she ended up having an infant born with a really serious heart defect. So these aren't things to be taken lightly. So another neurotransmitter I wanted to mention was GABA. That is gamma-aminobutyric acid. And this is an inhibitory neurotransmitter in the brain. I'm sure you've probably heard the term endorphins. Endorphins and enkephalins actually act in the central nervous system to block pain signals. You've heard of the runner's high. I've heard rumors of it. I'll never have one. So I've always said if you find my body on a running trail, you know I've been murdered. My body was dropped there. But supposedly when you are a runner and you can kind of hit that wall and you can keep on running through that, you have an endorphin release. And so I guess you don't feel like your body telling you to stop. So another thing I wanted to mention, aspartate and glutamate. These are excitatory neurotransmitters, and these are involved in memory and learning. And I wanted to kind of point a couple of things out. If you look at the term aspartate, you may realize that's kind of close to the term aspartame. And glutamate, you may have seen that in MSG, monosodium glutamate. So it's a little bit scary when we start adding these in our diet. And some of these have actually been indicated in memory loss in humans. Okay, let's finish up here and let's review this. We talked about this earlier. I told you if you've got your presynaptic neuron and now you've got your postsynaptic neuron. So that's number 1 and number 2 down here. And you'll be releasing neurotransmitters, which will be having an effect on that postsynaptic membrane. If the effect of that neurotransmitter is to depolarize that postsynaptic membrane, and again in the central nervous system it's often glutamate, 75% of the time it is, this is called an excitatory postsynaptic potential. So I've already told you, we talked about excitatory neurotransmitters and inhibitory neurotransmitters, and we know about action potentials, so let's put those terms together. So an EPSP, excitatory postsynaptic potential, is when this neurotransmitter that's coming from here is going to cause an action potential which will be transmitted down that neuron. Now, what happens if the neurotransmitter that's released from the presynaptic neuron inhibits or hyperpolarizes that postsynaptic membrane? That's going to be called an IPSP, an inhibitory postsynaptic potential. And again, that's often going to be the gamma-aminobutyric acid, the GABA I mentioned earlier. So an IPSP is going to make the postsynaptic cell less likely to have an action potential, and the EPSP is going to make the postsynaptic cell likely to have an action potential. One of the problems that we have when we start trying to teach neurology is that we talk about a neuron synapsing on a neuron. But in real life, you're talking about neurons that are getting input from thousands and thousands of presynaptic cells. Some of those presynaptic cells might be excitatory, some might be inhibitory. So what determines what that postsynaptic cell is going to do? And it's pretty simple. It's called summation and majority rules. If the postsynaptic cell receives more input that's inhibitory, it's not going to have an action potential. If the postsynaptic cell gets a lot more EPSPs, then it's going to produce an action potential. The word temporal means time, as in tempo. So with temporal summation, we're talking about a single synapse, and it's firing so often that it's going to be creating an action potential in the postsynaptic cell. So you've got one synapse that is repeatedly firing very quickly. With spatial summation, you're talking about many different synapses all over the cell. So let's say that you've got a neuron, and its axon is going way off up here, and you've got presynaptic cells that are firing all over this neuron. Let's say the red ones are going to be EPSPs, and let's go ahead and make some yellow ones be IPSPs. So in this case, because you've only got 3 IPSPs, and you've got 1, 2, 3, 4, 5, 6, 7, 8, 9 EPSPs, then the result of the spatial summation for this neuron is going to be excitatory, and so you're going to have enough to be able to produce an action potential in that postsynaptic cell. And basically, the very last slide of this, I just want to mention neural circuits. Again, we're so simplistic in how we picture neurons communicating with each other, and they actually communicate in electrical circuits. You can have a diverging circuit where you've got one neuron that's going to be synapsing on many neurons. You can have converging circuits where you've got many neurons synapsing on one. You can have reverberating, which is basically you've got a neuron firing on a second neuron, but then it's also reverberating back, so it kind of echoes on itself. Then you've got parallel after discharge, which is where you'd have one neuron that's going to be firing on many, and then it ends up going back to one. So again, this is much more complicated than what we've made it look like, but we have to start with some very basics. So this finishes the chapter on nervous tissue. We haven't talked about nervous anatomy yet, but with action potentials and summation and all these concepts, they're very abstract, and so I'm going to strongly recommend do your reading, read several sources if it helps, and then also go to YouTube and get some animations, because these are so abstract. Some animations can really kind of give you a light bulb moment. So I'll see you soon on the next one.